Imagine for a second that you are looking at a highly detailed, just an incredibly massive map of a bustling city.
Okay, I'm picturing it.
Right, so you can clearly see the towering skyscrapers, the tiny neighborhood streets, the sprawling parks.
The whole layout.
Exactly. But there is a serious problem. Because of a massive printing error, the bright, vibrant colors from those giant skyscrapers are, well, they're bleeding outward.
Oh, like the ink is running.
Yeah, the ink is literally spilling over into the small neighborhood streets, completely washing them out. It makes it practically impossible to see what is actually hiding in the shadows of the big buildings.
That sounds like a pretty messy map.
It is, and today we are looking at how scientists have identified a strikingly similar color bleed happening right now, but at the microscopic level.
Right, down in the actual biology.
Exactly. It's a distortion occurring when we try to map the cells inside human tumors. And more importantly, we are going to explore how a groundbreaking computational tool is finally cleaning up this image, to reveal the true cellular battleground.
And that color bleed is really the focus of our mission today. We are exploring a fascinating April 2026 paper from Nature Methods.
Yeah, it's titled Resolving Sensitivity, Specificity, and Signal Contamination in Xenium Spatial Transcriptomics.
Quite a mouthful, but it's an incredible look at the absolute bleeding edge of biotechnology. I mean, it covers the invisible errors that plague it and the brilliant mathematical solutions being developed to fix those errors before they just derail cancer research completely.
It's super important stuff. So to really appreciate what is going wrong, we have to understand what is going right with this technology. Let's set the stage a bit.
Good idea.
For years, if you wanted to know what a cell was doing, what genes it was turning on or off, you used a technique called single-nucleus RNA sequencing.
Right, and the industry standard platform for this is called Chromium.
Yeah, and I always think of traditional RNA sequencing like throwing a beautiful, complex fruit salad into a high-powered blender.
That is a great analogy.
You get a highly detailed readout of the resulting smoothie. You know exactly how many strawberries, blueberries, and bananas went into the mix.
But you've utterly destroyed all the spatial information.
Exactly. You have absolutely no idea if the blueberry was sitting right next to the strawberry before you hit the blend button.
And that loss of spatial context, I mean, it's a massive handicap for biologists, especially when we talk about cancer research.
Because location matters.
It matters so much. You desperately need to know if an immune cell is actively engaging with a tumor cell or if it is just sitting harmlessly on the completely opposite side of the tissue.
Which brings us to the new tech.
Right. That is the exact problem spatial transcriptomics was built to solve. Technologies like the Xenium platform map gene expression directly onto intact tissue.
So to use the analogy, they keep the fruit salad perfectly whole.
Yes, exactly. You get to see the transcripts, the RNA molecules that act as the physical messengers for genes, exactly where they are located in the tissue.
Okay, so I know this paper is based on a massive new data set. Just how big was the data set they used to test this Xenium technology?
Staggering. I mean, truly staggering. The team behind this research, Maria Billis, Daria Busta, Jonathan Back, Senbai Kang, and their colleagues, they built one of the most comprehensive Xenium data sets ever generated.
We're talking, what, a few dozen samples?
We are talking about 41 distinct tissue sections drawn from breast and non-small cell lung cancers.
Oh, wow.
Yeah, spanning 27 different human donors. And they didn't just look at a handful of genes either.
Right, because usually these panels are pretty limited.
Exactly. But they rigorously compared multiple targeted gene panels. They used a breast panel, a lung panel, a custom immuno-oncology panel, and the incredibly dense new 5K prime panel.
The 5K meaning, what, 5,000 genes?
5,000 different genes simultaneously.
That is just an unbelievable amount of data.
It is. And crucially, they also had matched blender data, the traditional Chromium sequencing, for those exact same samples to serve as a baseline comparison.
Okay, wait, hold on. I need to unpack this.
Sure, go ahead.
If Xenium is this highly advanced, high-resolution map that keeps the tissue perfectly intact, and they are generating this monumental mountain of data, why did this team need to dedicate an entire highly complex paper to studying technical noise and signal contamination?
It seems counterintuitive.
Yeah. If the technology is that good, shouldn't the data just speak for itself?
That is the million-dollar question, and it really gets to the very heart of the physical limits of our current microscopy.
Okay.
The researchers found that while Xenium is incredibly sensitive and highly reproducible, it suffers from a systematic glitch.
Like a bug in the software?
No, it is not that the machine is physically broken or malfunctioning. It is a fundamental physical and mathematical limitation of how we capture tiny strands of RNA in incredibly crowded biological spaces.
So it's just the reality of the physics.
Right. The researchers termed this phenomenon transcript spillover.
Transcript spillover. Okay, let's just pause and recap what we've built here. We have a revolutionary mapping tool that keeps cells intact, but it has a built-in distortion when things get crowded.
Spot on.
So before we get into how they fix it, how does Xenium even know what it's looking at? I mean, how does the machine decide where one cell ends and another begins?
It relies on a process called segmentation. Imagine looking down into a pitch-black room where you have dropped a glowing fluorescent dye.
Okay, I'm with you.
By default, the Xenium platform uses a chemical stain called DAPI, and DAPI specifically binds to DNA.
Which is in the nucleus.
Exactly. Because DNA is housed in the nucleus, DAPI lights up the center of the cell brilliantly in the dark.
But the outside is still dark.
Right. The camera only clearly sees that glowing nucleus. It does not inherently know where the outer wall of the cell membrane, is physically located.
So it essentially has to guess. It's like seeing the yolk of a fried egg in a dark room and having to mathematically estimate where the edges of the egg white are.
That is a great way to put it. The fried egg analogy is perfect. Because Xenium is essentially flying blind regarding the outer wall, the standard algorithm defaults to drawing a 5-micrometer radius expanding outward from the nucleus.
It just draws a perfect circle.
Basically, it draws a circle and says, everything inside this boundary probably belongs to this cell.
But cells aren't perfect circles.
No, they definitely are not. Now, for the massive 5K prime panel, they did attempt to use something more advanced called multimodal staining.
What does that look at?
This looks at the nucleus, the cytoplasm, and the membrane to try and draw better boundaries.
Oh, so trying to see the actual egg white.
Yes, but even with those advanced chemical stains, the computational boundaries were still deeply imperfect.
And I assume here is where the color bleed from our city map analogy happens.
You guessed it.
Because cells inside a real human tumor are not neatly spaced out on a grid. They are packed together in a frantic, chaotic environment.
Exactly. They are squished, overlapping, and constantly fighting for physical space.
Which means the RNA transcripts from one cell actually diffuse or spill over across those imperfect borders into the territory of adjacent cells.
Which creates artificial entities in the data that biologists call doublets.
Doublets, like two cells stuck together.
Well, in older sequencing methods, a doublet usually meant two actual cells physically got stuck together in the machine tube.
Oh, like a physical clumping error.
Right. But here in Xenium, it's an artificial, purely computational doublet.
Right, meaning what exactly?
It is a single distinct cell sitting on a slide that mathematically looks like a bizarre hybrid of two entirely different cells simply because it is wearing its neighbor's leaked RNA.
Let me push back on this for a second.
Okay, push away.
If the borders are just sloppy because the algorithm draws a 5-micrometer circle, why is the solution an entirely new mathematical framework? I mean, we have unbelievable AI right now.
We do.
Why not just use better image recognition to draw tighter, more accurate borders around the cells and stop the bleed?
It's a logical question. But the researchers actually proved it is not just a supple two-dimensional border issue that you can fix with a sharper pencil.
Why not?
First, you have to consider the z-axis.
The depth.
Yes. A slice of tissue is three-dimensional. It has depth. Cells are often stacked directly on top of each other in the tissue slice.
Oh, I see.
So when the Xenium camera takes a top-down image, the RNA from the cell living below literally bleeds up into the image of the cell above.
So no matter how perfect your circle is in 2D, the stuff underneath is ruining the picture.
Exactly. But even beyond that vertical overlap, there is actual physical diffusion of RNA in the horizontal plane.
Like the molecules are actually floating away.
Yes. The team proved this definitively by creating a metric called the spillover index.
Okay, I love when researchers put hard numbers to a theory. How do they actually prove these cells were stealing RNA from their neighbors, rather than just randomly glitching out?
They utilized a mathematical concept called cosine similarity to track the contamination.
Wait, cosine similarity? You completely lost me there. Can we get a translation on that?
Think of it like comparing two complex cooking recipes.
Okay, recipes. I can do recipes.
Cosine similarity doesn't care if you baked a massive wedding cake or a tiny cupcake. It just checks if the exact ratio of flour, sugar, and spices is identical.
Ah, so it's looking at the proportions, not the total amount.
Right. In this case, the researchers looked at the specific recipe of the foreign contaminating RNA found inside a cell. Then they looked at the recipes of the cells living physically next door.
Like literally touching them.
Literally within a 15 micrometer radius. They used cosine similarity to ask, does the specific blend of this contamination perfectly match the RNA recipe of the neighbor?
And I'm guessing it was a perfect match.
A perfect match.
If you look like you are mysteriously expressing a secondary identity, it matches the cells crowding you. It is like finding a blue ink stain on your shirt and realizing you are sandwiched on the subway between two people wearing wet blue paint.
That is exactly it. You didn't generate the blue paint, you caught the spillover.
So let's recap this section. We've got 3D stacking, horizontal leaking, and mathematical proof using recipe matching that cells are absorbing their neighbor's RNA.
Yes, and this spillover index revealed a very clear dynamic of victims and culprits in this microscopic environment.
Okay, who are the culprits?
The biggest culprits in the tissue are the malignant tumor cells.
Why them?
Because they are physically massive, highly metabolically active, and absolutely packed with RNA. They're basically roaring RNA factories.
And the victims?
The victims, unfortunately, are often T cells.
The immune cells.
Right. T cells are tiny immune cells that have very low natural RNA content to begin with.
Which sets up a terrible scenario. If a small, quiet T cell is sitting right next to a massive, loud, malignant tumor cell, the tumor's RNA completely overwhelms it.
Completely grounds it out.
To the Xenium machine reading the data, the T cell suddenly appears to be expressing tumor-specific genes. What kind of genes are we talking about here?
The paper specifically highlights genes like MUC1, FASN, and EPCSAM.
And
These are classic epithelial and tumor markers. When a T cell absorbs this spillover, suddenly your crucial immune cell, computationally, looks like it has cancer.
Which is biologically nonsensical. I mean, an immune cell doesn't suddenly turn into a tumor cell just by standing next to one.
Of course not.
So if we know the map has this physical glitch where crowded cells are bleeding RNA into each other, how do we actually clean it up? The researchers developed a tool called SPLIT. What does that stand for, and how does it fix them out?
SPLIT stands for Spatial Purification of Layered Intracellular Transcripts.
That is a very scientific acronym.
It is. It relies on a computational framework known as RCTD, or Robust Cell Type Decomposition.
Okay.
Now, remember that pristine blender data we talked about earlier, the traditional chromium single-nucleus RNA sequencing?
Right, the data that loses the spatial map but gives us a perfect ingredient list for each individual cell type.
Exactly. SPLIT uses that Chromium Blender data as a pure, uncontaminated reference dictionary.
Oh,
RCTD takes the blurry, contaminated cell from the Xenium map and mathematically models it as a mixture.
like unmixing the smoothie.
Sort of. It looks at the cell and says, this observed data is a combination of a primary cell type, which we'll call weight 1, mixed together with a secondary contaminating cell type, which we'll call weight 2.
I think an audio engineering analogy works perfectly here.
Let's hear it.
Imagine a recording studio. You have a quiet acoustic guitar that represents our tiny P cell.
Okay.
But the guitarist is sitting right next to a heavy metal drummer. That is our massive tumor cell. The microphone on the acoustic guitar is inevitably picking up the crashing drums. It is a contaminated audio track.
And because we have the chromium sequencing data, we have isolated tracks.
Right.
We know exactly what a pure acoustic guitar is supposed to sound like,
Sure.
and we know exactly what the heavy metal drums sound like. We have the pristine reference profiles.
So SPLIT looks at the messy, blended audio track from the Xenium map, compares it to the pure guitar track, and mathematically calculates the exact acoustic signature of the drum noise.
It's exactly.
Then it literally acts like a noise-canceling headphone. It subtracts the drums from the audio file, leaving you with a perfectly clean, purified acoustic guitar track.
It is a beautifully elegant solution. It models the expected fraction of transcripts coming from the primary cell and purifies the signal by entirely removing the fraction coming from the secondary contaminating cell.
That is genius.
But during their testing, the researchers encountered a fascinating and honestly dangerous edge case.
Oh, what kind of edge case?
What happens when the contamination is so extreme that the system gets confused about which instrument is the primary signal?
Oh, right. What if the heavy metal drummer is playing so loud that the microphone on the acoustic guitar actually thinks it is recording a drum kit that just happens to have a faint guitar playing in the background?
Exactly. The algorithm would accidentally identify the tumor cell as the primary identity and flag the T cell's natural RNA as the secondary contamination.
And if SPLIT just blindly went ahead and subtracted the secondary signal.
It would delete the true T-cell data and keep the fake tumor signal.
It would literally erase the immune cell from the map.
It would. To prevent this catastrophe, the team developed a nuanced, fail-safe feature called SPLIT shift.
How does SPLIT shift stop the algorithm from deleting the wrong data?
It makes the algorithm neighborhood aware.
Neighborhood aware, meaning it looks around.
Yes. Before purifying any cell, SPLIT shift looks at the physical environment surrounding it on the Xenium map.
Okay, so it checks the neighbors.
Right. If a cell is labeled as a tumor primary, but it is physically surrounded on all sides by T cells, and its secondary label is T cell, the algorithm flags a potential label swap.
Oh, I get it.
It reasons, wait a minute, physically this object is deep inside an immune cluster. It should be a T cell.
So it overrides the mistake.
It dynamically shifts the labels, making the T cell the primary identity and the tumor the secondary. Only then does it subtract the tumor noise.
It is literally like having a smart sound engineer who stops looking at the mixing board, looks through the glass into the studio, sees the guy is physically holding a guitar, and realizes the cables got plugged in backward.
That is the perfect way to visualize it.
They flip the switch before hitting delete.
It is a critical intervention that preserves rare, highly vulnerable cell populations that would otherwise be entirely lost to the background noise.
Okay, so SPLIT is clearly highly sophisticated. Let's do a quick check-in. We've got this noise-canceling algorithm that uses a clean dictionary to subtract neighbor noise, and it has a built-in safety switch so it doesn't accidentally delete the quiet cells.
That's the summary, yes.
But were scientists just ignoring this noise before this paper came out? I mean, how does SPLIT compare to the older correction methods that labs were already using?
The scientific community definitely knew this noise existed. No one was ignoring it. The paper rigorously compares SPLIT to older correction tools, specifically methods called overall pi and resolves.
And how did those older tools attempt to fix the color bleed?
Methods like overall pi often rely on rough techniques like vertical subslicing.
What does that mean?
Trying to just shave off the top and bottom of the data mathematically.
Oh, just cutting away the blurry parts.
Right. Other older models rely on heuristic corrections or highly complex, non-interpretable black box algorithms.
Black box meaning we don't really know how they make their decisions?
Exactly. But the practical outcome of these older methods is almost always the same. They end up forcefully deleting massive sweeping amounts of raw gene counts from the data set to try and clean the image.
So they are essentially using a sledgehammer to fix the problem instead of a scalpel. They just smash the data until the noise is gone.
Which is incredibly dangerous in science.
Because you're losing the actual information.
When you forcefully delete that much raw data, you destroy the statistical power of your entire study.
Right.
Imagine running a clinical trial and throwing out half your patient data just because your thermometer was a little dusty.
You'd get laughed out of the room.
You might technically clean the signal, but you have left yourself with far too little data to make confident, reproducible scientific claims.
And SPLIT avoids this.
Yes. SPLIT, by contrast, is a reference-based signal decomposition tool. Because it selectively subtracts mathematically modeled proportions of RNA rather than blindly deleting swaths of data, it preserves the gene detection levels beautifully.
You keep your statistical power while losing the noise.
Precisely.
That makes perfect sense mathematically. But I really want to elevate the stakes here for you listening.
Good idea.
We've talked a lot about the math, the algorithms, the cosine similarity. We have successfully fixed a math problem on a computer. But what is the actual biological payoff? I mean, why should a cancer researcher or ultimately a patient care that SPLIT exists?
The biological payoff is profound, and it all comes down to something called cell-cell communication.
Okay, break that down for us.
In modern biology, researchers use algorithms to study how different cells talk to each other across the tissue. We look for a cell expressing a specific signaling molecule and a neighboring cell expressing the matching receptor for that signal.
Like sending a text message and seeing the other cell receive it.
Exactly.
Yeah.
But if we have uncorrected transcript spillover, where the tumor's RNA is bleeding into the T cell,
your analysis software will falsely conclude that the T cell is naturally producing those tumor genes.
yes, and that it is participating in some complex, novel biological interaction with the tumor.
Which is completely false.
It creates a complete optical illusion of cell-cell communication that simply does not exist in reality. It is generating fake biology.
And this is where the implications become terrifying.
Very much so.
We are currently in the golden age of artificial intelligence. Biotech companies are feeding massive data sets into foundation AI models to discover new drugs.
Right.
If those AI models are trained on this uncorrected, bleeding Xenium data, we aren't just making a small error in one paper. We are actively poisoning the well.
The AI will start hallucinating these fake biological interactions and baking them into our fundamental understanding of human disease.
We could waste billions of dollars and years of research chasing ghost interactions that were literally just camera blur.
That is exactly the danger. The integrity of the data is paramount. And with the signal finally purified by SPLIT, the researchers proved just how much reality was being hidden by the noise.
What did they find when they cleared the smoke?
Once the tumor spillover was stripped away from the T cells, they were able to cleanly reveal specific T cell exhaustion signatures that were physically associated with the malignant cells.
What exactly is T cell exhaustion, and why is it so critical for cancer researchers to get that data right?
T cell exhaustion is a clinical phenomenon where the immune system has been fighting a tumor for so long and under such harsh conditions that the T cells basically burn out.
They just get too tired to fight.
They lose their ability to attack, they become exhausted. And tumors actually actively exploit this, sending out signals to force the T cells into exhaustion so the cancer can hide and grow unchecked.
Wow.
Identifying exactly where, when, and how T cells become exhausted in the tumor microenvironment is the absolute holy grail for developing new immunotherapies.
Because if you can find them, you can help them.
Right. These new drugs are specifically designed to wake those exhausted T cells back up so they can kill the cancer.
But before SPLIT was applied to this data, those vital clues, the specific genetic signatures proving the T cells were exhausted, were completely obscured by the screaming noise of the tumor cells' leaked RNA.
Yes, the immune cells look like tumor cells, so the delicate exhaustion signal was drowned out.
Only after SPLIT cleaned the audio track was the true, desperate state of the immune system revealed.
It provides oncologists and researchers with an accurate, high-fidelity map of the microscopic battlefield.
That is just incredible.
Without SPLIT, you are trying to strategize a war based on a map where the enemy's colors have bled all over your own troops, making it impossible to tell who is winning and who needs reinforcements.
That map analogy really brings it all home.
It really is a paradigm shift in how we process and trust spatial transcriptomic data. It proves that the high-tech image alone isn't enough. The rigorous mathematical interpretation of that image is where the biological truth actually lies.
Today, we went on a serious journey into the microscopic world. Let's just wrap it all up.
Sound good.
We explored the staggering scale of this new Xenium data set, 41 tissue sections, 27 human donors, thousands of genes mapped directly onto intact cancer tissue.
A monumental effort.
We diagnosed the invisible, insidious problem of transcript spillover, where massive RNA factory tumor cells physically bleed their signals into vulnerable immune cells.
Right.
We saw how sledgehammer methods destroy data and how the SPLIT computational method acts as a sophisticated reference-based audio engineer to save it.
By subtracting that spilled over noise.
Yeah, and using SPLIT shift to prevent catastrophic label swapping, so the algorithm successfully preserved the statistical power of the data.
Without losing the real biology.
And the payoff was just monumental, rescuing future AI from hallucinating fake biology and revealing the true genetic signatures of T-cell exhaustion in the fight against cancer.
It is a remarkable piece of science.
It is an incredible piece of scientific problem solving, but I want to leave you, our listener, with a final provocative thought to mull over as you go about your day.
Let's hear it.
For decades, our entire understanding of biology, of disease, of medicine has been heavily based on what we can visually observe under microscopes. Seeing is believing, right?
That's the old saying.
But if the SPLIT algorithm has just definitively proven that the cells in our absolute highest tech, most advanced microscopic images are actually wearing their neighbor's RNA clothes, what other established cellular interactions, what other unquestioned facts sitting right now in our medical textbooks are actually just optical illusions waiting to be mathematically purified?
And that is a question that should keep every biologist awake at night.
Thanks for joining us on this deep dive. Until next time, keep questioning what you see.